So I'm really happy to be hosting Dr. Désirée Schmitz to discuss her recent PLOS ONE Lab Protocol paper entitled A New Protocol for Multispecies Bacterial Infections in Zebrafish and their monitoring through automated image analysis, and the associated detailed protocol in protocols.io. Desiree was born in Germany and studied environmental sciences at ETH Zurich in Switzerland. She's a clinical microbiologist who, during her PhD at the Department of Quantitative Biomedicine of the University of Zurich, studied microbial ecology and evolution, focusing on interactions between bacterial pathogens and their impact on host organisms. She defended her thesis in 2023. Now, Desiree is a postdoc at Harvard Medical School and investigates antibiotic persistence and resistance in bacteria. Desiree is passionate about science communication, supporting educational equity, and staying active through pursuits like marathon running and kite surfing. Desiree, thank you so much for being here with us.
Thank you so much for inviting me. It's a pleasure to be here and to be able to talk about my article.
So to kick off, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yeah, definitely. So this protocol is about infecting 2-day-old zebrafish into a specific organ with one or multiple bacterial species, then monitoring the host survival as well as the bacterial infection inside the host. And importantly, it gives you all the tools to do like a mostly automated image analysis. So it consists actually of 3 different protocols. Protocol A is the zebrafish infection into the otic vesicle. The otic vesicle is the inner ear structure of the zebrafish. Then we have protocol B, where you embed the zebrafish and you image them at the microscope. And protocol C is the automated segmentation of the otic vesicle and the image analysis itself. Now, why is this relevant? We are avoiding research with higher animals like mice that would feel pain at this age, yet we're testing it in an organism that is already a vertebrate and it has innate immunity. And the cool thing is you can do both single and multi-species bacterial infections with the goal to improve our understanding of both the host and the bacterial side. And I think ultimately this can inform treatment approaches.
So to understand better the significance of these 3 protocols put together, can you tell us a little bit what was the state of the art when you decided to optimize the protocol? So how other people had studied multibacterial infections before in whole organisms?
Yeah, definitely. That's a very good question. So when I started out, most of the literature was on single bacterial infections. And the thing that was really missing, that was very clear to me after working on this for some time, is that there was not really a data analysis of multi-species infections into a specific organ in the zebrafish, let alone a semi-automated one. So when I started out, I really saw that as a gap. And I have to say, like, one of the motivations for me was also because I established the zebrafish as a model host in the Krumali lab, where no one had previously worked with zebrafish at all. So I got this challenging task to establish the zebrafish as a model organism, which was really fun and interesting, but it also meant I had to learn everything myself. I had to read the literature. I had to go and talk to people working with zebrafish and ask them how they do it. And through all this, I realized that there didn't exist a holistic protocol that combines infections into a specific organ, as well as the imaging, and then also the automated image analysis. And as a microbiologist, I'm most interested in the biological data, but there were many steps missing to get to that data. And I hope this protocol fills those gaps.
And this might be a tricky question because we're talking about 3 protocols, but what do you think are the most critical steps? Those that the users should be really paying attention to?
Yeah. So I think the thing that is most critical or that is most difficult about it is infecting the zebrafish correctly. This is the protocol A, the first part. And I mean, obviously other people in the literature that like have done it, other researchers have done it. But I think this is the most critical part, because if you don't get the bacteria inside the organ or too few, you don't have an infection. You cannot generate any data. You don't have anything to study. And there were obviously already protocols out there that did infections, but they seemed very tricky to me, at least in my hands. And the cool thing is I found Felix Ellett and Daniel Iremia from Harvard. They developed a wonderful mold to specifically infect 2-day-old zebrafish to make the infections easier. And with their molds, I could then streamline the protocol that was in place from others. So that really helped a lot. But I think really the most critical step are the infections. It really needs a lot of practice to do zebrafish infections, even with that streamlined protocol that we have published. That's the part that you need to spend most time on, in my opinion. Yeah, I think at least from when I compare it to the other model organisms that I've worked with, and during my PhD, that was mostly the larvae of a moth called Galeria melonella. Those larvae infections are very easy. Like, Gabriel, I could show you today. You would be able to do the infections tomorrow yourself, like without my help. For zebrafish, you need a lot more days to practice and practice and practice. So I think that's the most critical step.
And again, you know, we're talking about 3 different protocols, but for any of them, was there anything that as you were optimizing, you tried but didn't work and didn't make it to the final version of the protocol?
Well, there are always things that don't work. In the end, you try and try again. Most things did work in the end, but I think that maybe it's not something that didn't work, but something that I just didn't get to because of time constraints. I would have loved to continue with our setup to then investigate an infection in 3D, because obviously an organ is a 3D thing, right? And now the protocol looks at a 2D, like a section of that organ, and looks at the bacterial infection in that Z-stack, basically in that slice that you're looking at. But if you could go through the whole organ, you have like all Z-stacks in there. This could obviously tell us a lot more about the dynamics and the spatial aspects of a bacterial infection, especially when you're investigating multispecies infections. I do have the data for like all of my infections. I did like all the Z-stacks of the whole organ. But yeah, I had to finish my PhD at some point and had to wrap up to start my postdoc here at Harvard. But for anyone who's interested, you can also work with my data. It's publicly available. There's a link in the article. You can adapt the protocol and use it for 3D. That would be amazing.
And I am curious, why did you decide to focus on the otic vesicle?
That's a very good question. Yeah, so actually when I started out working on zebrafish, I tried all different locations for injections. I mean, into the bloodstream. I did bloodstream infections. I tried different organs. Why did I focus on the otic vesicle? Simply because it's a very nice and easy organ to work with, in the sense that for our purposes of imaging, it's really nice if you have one location versus having the whole bloodstream, because then you have to like image the whole zebrafish. This is on the one hand very time consuming, because you have to image the whole zebrafish. That just takes a lot of time. So having all this time and having to spend all this time at the microscope also means it's costly. Because I mean, at least in Zurich, we could book the core facility, but that costs money per hour. So I focused on the otic vesicle because of that, and also because it's a contained organ and we can like easily follow an infection in there. It's contained in there, which makes it really nice for data analysis. So basically I have my one field of view and I could then easily analyze my analyze my data. And that was very, very useful to start out with. But honestly, this could also be adapted for other organs of the zebrafish. You could definitely do this in other organs as well and adapt this protocol to them.
And would you expect different organs of zebrafish to respond differently to bacterial infection? And if so, how would you adapt the protocol to infect other organs?
I do think that different organs could respond differently to multibacterial infections or single bacterial infections. One of the main reasons I think is that some organs, as I said before, like the otic vesicle, for example, it's much more contained, so the bacteria shouldn't like easily escape. But for example, if you have an organ that's more easily accessible, then also the host's immune system, the host's immune defense can more easily get to the infection and clear it. So you have more neutrophils, more macrophages coming in and clearing the infection. So I do think you have to, there can definitely be differences depending on where you infect. If you would adapt the protocol, I think you could definitely, as I said before, you could adjust the protocol to other organs. That should work. Depending on the organ itself, though, I think it makes sense to think about the age of the zebrafish, for example. And this also means the mold might need to be different. So for this, you could contact Felix Elliott and Daniel Eremia at Harvard to see whether they have something that works for these ages, because that mold is really made to fit those 2-day-old zebrafish, and it works really well for that. But yeah, definitely you could do that.
Could the protocol be adapted to search for new antibacterial compounds or treatments?
Yeah, I think so. It could certainly be used to test potential new treatments. In terms of how to do this, I think you could adjust the protocol, for example, by including another injection step. And this injection could be at any time point. It could be before injecting the bacteria. It could be after injecting the bacteria. Any time point before you embed and image the zebrafish would be fine, I think. It really depends on the question you're asking and whether this requires infecting earlier, adding the treatment earlier, or after the infection. Yeah.
And so we've been discussing the protocol itself, but I would like us to change gears a little bit and discuss about your motivations to send a lone methodological paper.
Yeah, well, Gabriel, I have to tell you, I looked at so many different journals and types of methodological papers that they offer. And what fascinated me personally about PLOS ONE and protocols.io was the combination of having an actual scientific paper, but then also having a step-by-step protocol that any researcher can use to investigate bacterial infections in zebrafish, in my case. Yeah, establishing a new model organism in a lab is a lot of work. For me, that was the case. And I felt like, what can I give back from this experience to my peers, and how can I make their lives easier? Basically, I was thinking, well, if I had to start over again, this protocol would be so helpful for me to really establish a new model host in a lab that has never worked with zebrafish and has never infected zebrafish. So I really hope that this protocol-based article can be useful to other researchers.
How did the feedback that you received from the peer reviewers shape the final version of your protocol?
I think it made them clearer. So I have to say there were minor things that I adjusted regarding my protocols, but in the end, I think it should have made them clearer. Yeah.
And why did you decide to make the peer review history public?
Well, generally, I think you can learn a lot from looking at the initial paper and then how the peer review helped to improve that paper. So I think that is very useful for any person, for any researcher out there.
It is really inspiring to be talking to such an enthusiastic early career researcher. And this question might be a little bit speculative because the paper is relatively young, but what do you think will be the significance of having published the protocol and the paper for your career?
Mhm. Well, maybe I can first take like the personal view, and at least for me, this publication was not only the last of my 3 publications from my PhD, but it allowed me to bring everything together I had learned throughout my PhD and working on zebrafish. So it was a really nice conclusion of my work there. I also very much appreciated the collaboration with Tobias Wessler, with Hongwei Li, Björn Menze, and with my previous supervisor, Rolf Kümmerli. Everything went very smoothly, and I learned a lot from this. So I'm very appreciative of that. Then maybe looking into the future, I think it's also, I think it's great that I can say I don't just have like scientific articles published, but this combination that I really like, where you have a scientific paper, but also the protocols related to it, so that other researchers can make use of it for their research. So I hope that this can be helpful for others. And I think it's very cool to have this combination also.
So, Desiree, I don't have any other questions. I don't know if there's anything that you would like to add that haven't been discussed so far.
I think we covered most of it. I just have to say, I want to say that this process, like also working with protocols.io, worked very smoothly for me. And I really appreciated how I had to like come up with like my protocol, how working with this protocol-based structure also made like everything much clearer. And I had to like really think about like which information is needed and all of these things. And yeah, I can just say that it was a very nice work, in the sense also of like publishing this paper with PLOS ONE and with protocols.io.
Thank you.